It's the 18th of October, 2024, and this is the Room Now podcast. Hey, everyone. Hope you're having a good week. This week, we're going to talk about chatbots, GWAS, ANAs, D2T, PSA. It's a lot of letters and nonsense. Chatbots. AI is taking over. I don't know if you're using it. I use it a lot, actually. I think it's a great tool, and I think if you're not using it, you're missing out. The question is it, society going to hell in a handbasket, or is it change that makes you uncomfortable? There was a nice report this week that tested the ability of an artificial intelligence chatbot from Bing to make up information and answer questions about 50 of the most common drugs that are out there. So it provided 500 answers, and these AI-generated answers were all really very good. I mean, if you look at the actual accuracy of what was printed, it was over 90%. The problem with it was, however, one, it was not very readable, meaning it wasn't for the common man. Usually that's meant to be at about an 8th grade reading level. This was sort of written to complement the people who wrote it. So it was much higher than that. And then there were occasionally inaccuracies that the authors of this paper took the, extra leap of saying these were potentially life-threatening and serious complications to patient medication safety information. Again, a little bit of a stretch. By and large, AI was pretty darn good. What you need to know about AI is that we're really in the first or early generations of AI. It's getting better year by year, and its utility in practice is going up. I think you should be looking into it. The FDA did approve a new drug this past week, although it was a biosimilar for Stelara. I think that makes the fourth ustekinumab biosimilar to hit the market. This one's called Amyloidosa. Gotta say it 3 times fast. And it's ustekinumab in 4 letters that mean nothing. Again, it goes for all the indications of ustekinumab. That would be psoriasis, psoriatic arthritis, Crohn's, and ulcerative colitis. The originator sales for the J&J Janssen made product Stelara in 2023 was almost $11 billion. Not surprising that there's now four biosimilars for ustekinumab. Will that change things? It certainly has, I think, for the TNF inhibitors, especially for adalimumab, although the number one maker or seller of adalimumab right now still is the originator, Humira. The question is, will the overall cost of adalimumab and ustekinumab go down? By and large, it should. And will more people be treated because of the lower cost? I think it will be. Data starting to accumulate to that effect. A report this week on looked at cardiovascular health. It's data from the NHANES study and shows that the calculation of a cardiovascular health score, this means like blood pressure monitoring, lipid monitoring, control of glucose, things like that become cardiovascular risk factors. And if you get a score, and if you score well on this, for every 10-point increase in the CVH score, you had an overall significantly lowered risk of mortality. So as your cardiovascular health scores went up, meaning good things, your cardiovascular mortality went down, and it went down by 28% for every 10-point increase in this analysis of almost 6,000 studies, 6,000 patients. Most of the patients in the study were degenerative and osteoarthritis, degenerative disease and osteoarthritis, which is where all the benefit was seen. They didn't show statistical significance for inflammatory arthritis, but I think that was a sample size issue. Because certainly we know that when you have healthier lifestyles with inflammatory disease, patients do better as far as cardiovascular outcomes. Sticking on the cardiovascular outcome angle, why not look at European Heart Journal? I know you read it all. I do. This is a report from a group that's interested in RA and cardiovascular outcomes, and they specifically looked at cardiometabolic factors in RA patients who were pregnant. Overall, they found cardiovascular risk factors going up in RAs who became pregnant. You would think that would be relatively young RAs, right? 20 to 45. But in the last 15 years, the incidence of these things that we would call what? Hypertension, diabetes, gestational diabetes, obesity, and dyslipidemia. That's all gone up significantly in the last 5 years. Moreover, the RA patients that were included in the study tend to be a little bit older than the matched population, 31 years versus 28 years. And with age comes some of these cardiovascular risk factors. Overall, though, again, women who were pregnant included in this analysis had a higher risk of cardiometabolic risk factors and, consequently, more adverse pregnancy outcomes: preeclampsia, cardiomyopathy, arrhythmias, acute kidney injury, venous thromboembolism, and hospitalizations. So again, we think of our RA patients who get pregnant as being, they should be generally healthy, but, they bring a lot of RA to the table, and that means a lot of comorbidities. This needs to be considered really when you're counseling these patients. A large GWAS study of 2.6 million people looked at genes associated with gout. This GWAS study, I think the number was like 3/4 of the data came from 23andMe sample submission. From 2.6 million, they think they had 120,000 patients with prevalent gout. They detected 377 loci, 410 independent genes. About 150 of those were previously unreported that were associated with gout. This included genes associated with inflammation, epigenetic remodeling, cell osmolarity. I don't get that. NLRP3, the inflammasome, I get that. And clonal hematopoiesis, an increasing factor in disease risk for many of the autoimmune and rheumatic inflammatory diseases. But the bottom line on this, the takeaway that you'll see in the press was, gout is not so much a nutritional disease as most people think. It's very much a genetic disease. And I don't know that surprises you, but that's what is in the news this week on gout. Based on this large GWAS study and a large number of genes associated with inflammation and NLRP3 and CHIP, we do see the evidence that gout is very much genetically based. Another study that came out this week, which I would say is a kind of a no-brainer and a duh, but I reported on it anyway for reasons that it's boring. It's about biosimilars. I'm hard-pressed to find biosimilar data that's interesting, exciting, new, novel, gives you a ooh, wow kind of aha moment. And that's not one of these. This is a study of the biooriginators and biosimilars. The retro study with 2 years of follow-up showed that when you compared the patients who were taking either the originator or the biosimilar of either infliximab or etanercept, that serious infection rates were the same. Now, is that exciting? No, it's quite boring. But then again, I've been preaching for a while, boring is good. Boring is what we want in the outcomes of our patients. Live for boring. Lifestyle, exercise, diet, how you take your medicines, how you get your follow-ups, that should all be really boring because that's the key to success. This data, which is boring, is another added factor to success. So they looked at infliximab. All right, first let's look at etanercept. The rates of SIE, serious infection events, with etanercept were significantly lower than with infliximab, like 11.8 per 100 patient years. Actually, I think this is 1,000 patient years. And it was the same with the originator versus the biosimilar. It was 11.8 versus 8.4, not statistically significant between them. That's with etanercept. Infliximab SIE rates were almost 3 times higher, 33 versus 29. The originator, Remicade, versus the biosimilars, Inflectra and the other versions of infliximab biosimilars, 29 per 1,000 patient years. Not significant. Again, it's duh. I would expect that, but it does give you confidence when your patients are voluntarily or involuntarily switched to either an etanercept or infliximab biosimilar, that the outcomes are going to be the same as if they stuck with the original drug. I believe that strongly. Another meta-analysis this week looked at the risk of ILD. I was always perplexed by data that said RA patients that go on leflunomide have an increased risk of ILD. Really? RA patients that go on TNF inhibitors have an increased risk of ILD. I don't think so. And what about RA patients that go on the risk of any new medicine, including methotrexate? Increased risk of ILD. I think that's guilt by association. There's confounding going on here. So this particular study, a meta-analysis of 40 patients, 40 studies, almost 500,000 patients, almost 4,000 cases of incident ILD, basically showed, 1, no increased risk of ILD when starting a new DMARD. No increased risk of ILD when starting either tofacitinib or a TNF inhibitor. Great data from the oral surveillance study, which showed an odds ratio with either of them about 1 or 0.94. More importantly, they showed that methotrexate use did not in any way increase the risk of ILD, but in fact significantly lowered the ILD risk to the odds ratio 0.49 with confidence intervals of 0.32 to 0.76, a 50% lower risk when you compare patients that went on methotrexate versus those that did not. I think, again, this is boring but exciting, brand new, helping you to practice data. What is a lot of your practice? A lot of your practice is the ANA positive consult. Oh my goodness. I firmly believe that ANA positive arthralgists that come to me are not all going to be normal people. I believe about 5, maybe as much as 7, 8% of them will eventually become an autoimmune, inflammatory, or lupus-like disease. But that means 95% of them are not. So here's a study, a 2-year study of 207 asymptomatic ANA positive people. ANA positive, asymptomatic. They don't even have arthralgias. How many of them progressed to a systemic autoimmune rheumatic disease? What would be your guess? I told you my experience is that I just said ANA positive arthralgias, 5%. In this study of 207 asymptomatic ANA positives, they didn't go into how high the titer was. 11% progressed to a diagnosable systemic autoimmune or rheumatic inflammatory disease. That means 89% did not. Of the ones that did progress, almost half of them were lupus, 22% Sjogren's, 13% scleroderma, and 4% RA. So, again, this is, I think, comforting to know what the fate of your ANA positive consult is going to be. There was a report this week from, I think in J Room, on difficult to treat RA, testing 2 different criteria for not difficult to treat RA, difficult to treat PSA. Difficult to treat RA criteria were established by EULAR. There are 2 criteria that have been established by Perota and Kumthekar, and they compared them, and they have a sensitivity of about 35%, a specificity of about 85%. They both perform equally well. In both of these studies, somewhere between 15 and 20% of patients from a cohort of PSA patients qualified as having difficult to treat PSA. It's an important number to know because these are the people that you're really going to have to work at. These are the people that you're going to have to decide, is this a difficult to treat patient because of ongoing inflammatory activity? Some of them are. Most of them are non-inflammatory activity, which means that they need attention to fibromyalgia, other comorbidities, depression, pain due to mechanical causes, etc. So I think this is important. You'll be seeing difficult to treat reports and studies in the future. I think that's going to be really important for future research in this area. Speaking of psoriasis and psoriatic arthritis, an analysis of psoriasis patients, a sub-analysis of a deucravacitinib study. It was a phase 2 study of almost 250 patients with psoriasis, where they were treated with deucravacitinib for 12 weeks. They found that when looking at biomarkers, that they mainly came from the IL-23, IL-17 pathways, and that using deucravacitinib was associated with significant reductions in IL-17A, IL-17C, beta-defensin, PI3, and all of these correlated with changes in the skin activity score, the PASI score. Again, lending some credence to the rationale behind deucravacitinib use. We did talk about this a little bit last week, did we not? About why maybe ustekinumab might be an ideal target when you start looking at outcomes in this manner. A study of GI abnormalities in scleroderma, a prospective scleroderma cohort, found that GI tract disease was very common, almost 50%. Negatively, of scleroderma patients will have GI involvement of various types, and it will negatively impact quality of life, physical function, and their employment. But interestingly, no effect on mortality. Good to know. Zoster vaccination. Yes, our patients should be getting zoster vaccination, especially when they're immunocompromised, on JAK inhibitors, or over the age of 65. This is a VA study that looked at the use of the Shingrix vaccine, the recombinant zoster vaccine, approved in 2007, originally approved for adults over age 50, modified in 2021, and that was in 2017. In 2021, it was modified to include adults who are also immunocompromised, should be vaccinated with Shingrix or the recombinant zoster vaccine. In this veteran study, 90,000 veterans receiving immunosuppressives, only I think it was 23,000 of them actually received the zoster vaccine. So if you looked at the people the vets over 50 who were immunocompromised, only 36% received the recombinant zoster vaccine. That was at 2017. When the guidelines changed, the number went up from 36% to 50% were being vaccinated by 2023. So it did go up. It's still only 50% of the people who were in fact eligible. What about the vets who are under age 50? Again, at the original indication, only 3%. But then again, it wasn't indicated. But after 2020, it's only risen to 13% in younger people. The bottom line is we're not doing a good job of vaccinating against zoster with this inactive, very safe vaccine. Those who are unlikely to receive the recombinant zoster vaccine were males, African Americans, non-whites, non-urban livers. Interestingly and thankfully, RZV vaccination was higher in those receiving targeted synthetics, JAK inhibitors, biologic DMARDs, or combinations of drugs that either involve biologic or DMARDs or targeted synthetic DMARDs. So that's encouraging, but we can do a better job. 2 more reports this week were about the unconventional treatment of people with back pain. Both of these were published on MedPage today. The first one was from the British Journal of Sports Medicine. This is about the use of a run-walk program to treat people with chronic low back pain. They looked at adults who were under age 45 with chronic low back pain, and they compared, in this British study, those who received a structured program versus no program, those who were on the wait list for the program. And what was the intervention? Again, it was a formal education with digital and phone follow-up, I believe. And they showed in this 12-week run-walk program that the intervention had a significant reduction in pain intensity by, it looks like a median of 15 points. That was highly significant at p.003 compared to those who had no intervention. So this is just a run-walk program, right? No drugs, nothing else. Similarly, another study published this week about, I think this is from JAMA, looked at 216 patients with chronic sciatica due to herniated discs. And they were randomized to either receive sham acupuncture or 10 sessions of acupuncture over a 10-week period. Everybody's blinded. The assessor, the statisticians, the patients, even I think those who were given the acupuncture sham or real treatments. Well, actually, they couldn't have been blinded, right? So the bottom line was the results were significant and very significant for acupuncture. I notice this when I look at all the treatment guidelines for osteoarthritis of the knee, for instance, and hip, that many of the things we use don't work very well. But a lot of these alternative measures, like acupuncture, like chiropractic, like, in this case, a run-walk program, are highly effective. As we talked about last week, mindfulness therapy, highly effective. We need to use more of this. That's it for this week's podcast. Enrollment and registration is open for Room Now Live 2025, early February in Dallas. You can be at home. You can be in Dallas. It's going to be a fabulous meeting. Artie Cavanaugh and I are just putting it together. You're going to love it. See you there.